go into more details about nucleic acids. It is now known that there are two types of nucleic acids found in a cell. The deoxyribose nucleic acid, that is abbreviated as DNA, and the ribose nucleic acid, that is abbreviated as RNA. As I was mentioning earlier, as early as 1869, Miescher had identified a substance in the nucleus. He called it as nuclein. Some worker later found that this substance has an acidic property, called it as nucleic acid, and later it was found that such a substance not only occurs in the nucleus of a cell but also in the cytoplasm of a cell, and thereafter these two substances were identified. Though in 1869 itself Miescher was able to mention the name nuclein for the nucleic acid isolated from nucleus, a clear idea about the chemical nature of this substance could come only in 1953 with the works of two English biologists, Watson and Crick. They conducted a series of experimental investigations using X-ray diffraction and other sophisticated technologies, and were able to describe the exact chemical nature of DNA. Before going into more details about DNA, let us generally know about the nature of these nucleic acids. The nucleic acids are polymers. They can be described as polymers because they are composed of a large number of repeating similar units, and these repeating similar units are called monomers. The monomers of nucleic acids are called nucleotides. Therefore, both DNA and RNA can be generally described as polynucleotides. A nucleotide is a compound which is formed by a combination of three compounds. One is a sugar molecule. Let us abbreviate this sugar molecule as S. This sugar molecule is a monosaccharide and it is a pentose sugar, 5 carbon sugar. There can be two types of pentose sugar. One is ribose. Ribose is a typical pentose sugar. With the formula C5H10O5. C5H10O5, and this is the sugar present in RNA. Hence the name ribose nucleic acid. The other form of this sugar is deoxyribose, where one oxygen atom is less. So it is C5H10O4, and this is the sugar that is present in DNA. Hence the name deoxyribonucleic acid. Another compound present in the nucleotide is a phosphate. Let us symbolically abbreviate it as P. Phosphate is formed by orthophosphoric acid. H3PO4, and it is similar in both DNA and RNA, and it is this phosphate which is responsible for the slight acidic nature of these compounds, and hence the name nucleic acids. The third component in a nucleotide is called nitrogen base. Let us represent it as N. A nitrogen base is a basic compound containing nitrogen, an organic compound, basic in nature, containing nitrogen. It is a ring compound. It is of two types. One class of nitrogen bases are called purines. Purines are double ring compounds. There are two of them, One is adenine, the other one is guanine. Adenine and guanine. Let us abbreviate adenine as A, guanine as G. Both adenine and guanine occur in DNA as well as RNA. So they are found both in DNA and RNA. Another type of nitrogen base they are called pyrimidines. Pyrimidines are single ring compounds, and they are of three types. One is cytosine, let us abbreviate it as C. Like adenine and guanine, cytosine is found both in DNA and RNA. Then thymine, let us abbreviate it as T. It is found only in DNA. This pyrimidine is found only in DNA. And uracil, let us abbreviate it as U. Uracil is found only in RNA. In other words, purines, adenine and guanine occur both in DNA and RNA. Among pyrimidines, only cytosine occurs both in DNA and RNA. Thymine occurs only in DNA. Uracil occurs only in RNA. Therefore, DNA is composed of nucleotides of adenine, guanine, cytosine and thymine, whereas RNA is composed of nucleotides of adenine, guanine, cytosine and uracil. Now you can generalize and write like this. You can generalize and write like this. This is a symbolic representation of a nucleotide. P stands for phosphate, S stands for sugar, N stands for nitrogen base. So this is a nucleotide. This N can be A or G or C or T or U. We will go into more details about this.